Hey there, guys. So we're going to do a little bit more on bacterial genetics and think about some of the next steps in terms of DNA replication and the transfer of genes. Where we last finished off, we were looking at an electron micrograph, and I have to apologize I haven't posted the PowerPoints, but before I post this lecture, I will get them to you. And we can see that there are these different types of DNA molecules. We've got the chromosomal DNA, which is really quite long. I mean, these gigantic amount of strands, looks like spaghetti, but it is one large, gigantic twisted up ball of yarn. And then we've got plasmids, which are these circles. And the plasmids can be in different sizes as well, but generally they're not much above 200,000 molecular weight, or 200,000 nucleotides, whereas the genome is going to be in the multi-millions. So plasmids are a good bit smaller. Now, as far as DNA replication goes, it is broken up into a couple of different steps. The double helix is going to open up at a specific spot in the genome that is known as the origin of replication, or the ORI, the ORI. And DNA polymerase begins to fill in the spots. Now, the genome will have multiple origins of replication. And so the fork, as it is, will go until it either gets to the end of the chromosome or until it meets another fork. And we've got these two strands, the parental strand over here and the newly synthesized strand over here. And there's some interesting stuff in terms of how these things are working, and that one strand is actually going this direction and the other strand is in fact going this direction, but that's not all that critical for now. So the plasmid replication is actually working in a slightly different way, in that rather than having multiple origins of replication, there's a single one. And we end up starting here and then making what's known as a phi, P-H-I, the Greek letter structure. See, kind of with the O with the cross through it. And the replication continues, and then you end up at the termination of replication, and you seal this guy back up, and you make two different molecules here. So now, another critical part to the next step is taking the genome and transcribing it, and that is turning it into a gene. And I'm going to do this on the board just because I like working on the board. So here, let me crank you down a little bit and bring the screen up. So when we're thinking about DNA and drawing it and stuff, the fancy diagrams show it as this double helix, but it's really not very convenient to draw it that way from a cartoon structure or a cartoon perspective. And so I typically draw it as two parallel lines. All right, so it works like this. We've got the double-stranded DNA, and there's going to be a nucleotide that is known as +1. That's the first nucleotide of transcription. And what's happening is the RNA polymerase, the enzyme that is able to actually generate an RNA molecule, known as RNA polymerase, it's able to physically melt apart the double strands and actually generate what's known as a transcription bubble of about 50 or so nucleotides. It actually is going to open this piece up. And the RNA polymerase is actually reading this bottom strand. And as it progresses, it's making a RNA that's going to come wobbling out. Now, not to get overly detailed, but this RNA is identical to the top strand. the one difference is RNA has the base uracil instead of thymine, but in terms of sequence, it's really not all that important. So you make an RNA transcript. You have a transcription start site. That's one word. Transcription start. And then you'll have some kind of termination, a stop for the transcript, and you generate now an RNA. The next step is the RNA is going to be translated. And what this is going to do is turn this RNA sequence into an amino acid sequence. And the amino acid sequence is read 3 nucleotides at a time. 3 nucleotides are what you read to pick up determine one amino acid. And there is a set of structures. We have what's known as the ribosome, which comes in these 2 parts. It's a little bit like a hamburger bun. The small subunit and the large subunit of the ribosome. So, kind of have a list here. We have to have the ribosome. And then another thing we have are these adapter RNA molecules that are known as transfer RNA molecules. tRNAs, they actually have sort of a cloverleaf shape to them. They look a little bit like this, or a cruciform shape to them. And the amino acid is coupled to one end. And they will base pair at that spot. It's referred to as the codon-anticodon interaction. And the tRNAs then will be floating around in here. The next tRNA will come in. And then there is enzyme activity in the ribosome, like catalytic activity, known as peptidyl transferase. It makes a peptide bond. And it pops the amino acid from this tRNA off and links it to that tRNA. And then this one floats away. The whole thing moves down, and we have another 3 nucleotide slot for the next tRNA. And this continues on and on and on until you reach what's known as a stop codon. And you then have a protein that can float away, and the transcript can be producing multiple proteins, because as soon as the first ribosome complex clears the first part of this RNA, another ribosome can jump in. And so the same with RNA is that as soon as the RNA polymerase clears the transcription start, another RNA polymerase can come in. And so we're making multiple copies of RNAs, and then each copy of RNA can produce multiple copies of a protein. So you can go from a silent gene to a lot of an enzyme in like 10 minutes. I mean, it really will crank. So this is the mechanism by which we can generate a protein. And this leads us to what we think of as the bottom line for the central dogma of genetics. And let's just throw the slides back up, because now this is a rather brief introduction into the intricacies of protein transcription and translation. If you'd like to have more discussion, holler. This is what I do. This is my jam, as we might say. And the central dogma goes like this. It is the fundamental way that information is transferred in all organisms. Viruses may actually have an RNA genome that they can go into DNA, but other than that, we all have this fundamental instruction set known as our genome. Bacteria have it, plants have it, fungi have it, we have it. All creatures on the planet have DNA. And the DNA can be replicated. So DNA directs production of its own self. It copies itself, and that's known as replication. Then the DNA can also serve as the template for the production of a small piece of itself in the form of RNA. This is referred to as a DNA-dependent RNA polymerase, and we produce an RNA. The RNA can direct the production of proteins based on the RNA, and consequently, changes in the DNA can be reflected in changes in the RNA, and that can be reflected in changes in the amino acid sequence. So, this is translation. So, what this means is we are our genes, that our genes direct what our protein structure is going to be like. And hopefully you remember back in the early weeks, I was jumping up and down about the structure of a protein is critical to its function. And that structure is based on the amino acid sequence. The amino acid sequence is based on the nucleotide sequence. And the nucleotide sequence can be subtly altered to change different structures in the protein. You can have a whole range of proteins that are all functional, but functional in slightly different ways. And that's what gives rise to all the differences we see within a population in bacteria or people. We can also have changes in the amino acid sequence, or in the nucleotide sequence, that result in a non-functional protein. This would be a rather serious mutation, a deleterious mutation. So we've got RNA, or DNA being replicated. The DNA is being transcribed. The RNA that is produced is being translated. And we produce all kinds of different proteins. And these proteins are going to be what drive disease. Everything from endotoxins, which are going to cause toxic shock and edema and all kinds of things. Exotoxins, toxins that are being released. This is a great way to give yourself diarrhea, tetanus, paralysis, all kinds of things. We have polysaccharides that are being generated to produce the capsule, and that can prevent a bacteria from being phagocytized by the immune system. We can have the pili being produced that are going to anchor the bacteria to some part of the host. And this is what's going to be responsible for inflammation and necrotic tissue as you have a bacteria that's not being washed away but actually hanging off in one spot. We have all kinds of digestive enzymes that can begin to digest parts of your patient. Something obviously you may have heard of is drug resistance genes. Drug resistance means that the antibiotic is no longer functional. It's either being destroyed or its action is being blocked, and the infection is going to remain. The antibiotic is no longer functional. So mutations we can define as any change in the sequence of the DNA. Now, what this means is that there can be mutations that might be in a gene, and that could be deleterious or not. We can also have mutations that are outside of genes, and that may not have any effect at all. Just sort of a philosophical question, we can have mutations that don't really change anything, but there are ways of identifying individuals within a population. And is it a mutation if it doesn't do anything? In our definition, any change is a mutation. So we can think also about mutations as being either spontaneous, they are occurring through errors in DNA replication or errors in transcription. Transcription errors don't happen that much, but DNA replication errors, they happen not a whole lot. There's a lot of proofreading that goes into the system to prevent that from happening. Another kind of mutation, though, is an induced mutation, a mutation that is generated by some kind of external material. So we've got these naturally occurring mistakes, and then mistakes that are being kind of generated, enhanced by mutagens. Now, pretty much as a rule, all carcinogens are going to be mutagens. And pretty much all mutagens have the possibility of being a carcinogen. And that cancer results from mutations that occur. These mutations could be passed from one generation to the next, which generally the way cancer kind of works is if it's an inherited trait, it's going to be an inherited susceptibility rather than an inherited cancer. So you may have heard about the BRCA1 gene, a gene that's involved in breast cancer susceptibility. It happens because one of the copies of the BRCA1 gene has been mutated, and so the individual only has one functional copy. And if you're missing one copy, the chances of losing the second one are greatly enhanced. It's called a two-hit hypothesis. If you already got one hit, the chance of getting the second hit is really high. And that can lead to familial inherited cancers. Other things that can happen is being exposed to chemical mutagens. And they don't have to be chemicals. They can be gamma rays or UV radiation. Anything that can damage the DNA, these are things that can have effects. Now, mutations can be categorized a couple of ways. We can have a silent mutation. And this is going to be a mutation that doesn't affect the organism. And there's lots of DNA, less so in bacteria, but in us, we've got a lot of DNA that doesn't do anything. It's just got a spacer, it's just filler. And you can pick up mutations in that, never know it. Bacteria are a bit more compact, but even still, because of the way the genetic code works, you can change a nucleotide and it won't change the amino acid that nucleotide sequence is assigned or is assigning, and so it's a silent mutation. Another kind of mutation, though, that is more deleterious is what we call a null mutation. This is not a mutation that's silent. This is a mutation that knocks that gene out. The mutation is going to destroy the gene activity. It could either be a deletion of the gene or a mutation very early on in the RNA such that transcription is able to be carried out, but translation stops. You don't make a protein. Null mutations, these are the serious ones. A new trait. New traits are kind of rare, actually, but this is what we think of is a mutation is going to change how a gene works and make it so something else happens, something new happens. Now, hopefully, it's continuing to do its job, but maybe it does its job a little bit better, or it doesn't do its job quite as well. It's a little bit faster, a little bit slower. There are some kinds of mutations that can happen that don't have an effect too much on the functioning of the body or the organism, like hair color. You know, you can change hair color and not have a dramatic effect on the individual. So these are traits that can be developed. So virulence can be affected by mutation because a mistake in one generation is going to be a permanent change in the next generation, and that permanent change now can lead to another kind of change, or another mutation, and you can begin adding and building up mutations. And one base change can affect or will affect the RNA, and then that RNA might then generate a new amino acid sequence that it codes for in translation. So in the case, something you may have heard of is sickle cell anemia is a good example, that the normal hemoglobin has a glutamine at a specific spot. If that glutamine is changed to a valine, a different amino acid, it actually causes the hemoglobin to change shape and generates the sickle cell anemia traits, which is actually during stress, the red blood cell changes its shape and it can't actually pull oxygen in very well. And this is the result of a single nucleotide mutation. So you have the nucleotide sequence, the codon CTT. If you change it to CAT, CAT, the messenger RNA is GAA, and now the messenger RNA is GUA. That is going to change the overall amino acid sequence from a glutamine to a valine. So single nucleotide mutations, they can be very deleterious. So mutations occur randomly, just in the population. As organisms are carrying out DNA replication, errors happen. And they also can happen if organisms have been exposed to different kinds of mutagenic substances that are just in the environment. So the real point is mutations are going to happen. It's the selection for those mutations that ends up being really an interesting game. So you can have a mutation, but if there's not a selective pressure to kind of push populations towards that mutation, it's a mutation that happens, but it's kind of maybe being swamped out by everybody else that didn't get mutated. Because sometimes maybe the mutation causes that organism to still be alive, but not do quite as well. But if it does provide some kind of a selective advantage, say you are resistant to an antibiotic, until you're exposed to that antibiotic, that mutation didn't do anything for you. In fact, it might have screwed you up a little bit. But once you're in the presence of that antibiotic, woohoo, it is your time to shine, because baby, you are set to thrive, and everybody else can't make it. Now, these are the exception, because most mutations are going to be deleterious. But occasionally, you can pick up a mutation that, under the appropriate environmental conditions, gives you a selective advantage over the rest of those poor suckers that didn't pick up the mutation. So, if we've got mutations that are already present, we can expose bacteria to antibiotic, and then the bacteria are already present there, and they're going to continue thriving. If you've got a bacteria that is resistant, mutations can be induced, but you're not going to see these colonies until the bacteria are being selected for with antibiotics and things. So what this means is that random mutations are going to be generating resistance before exposure to antibiotics. But then we can have environmental directed mutations that are going to be caused by the exposure to that material. It's kind of an esoteric point, but not all that relevant. Part of what this means is that antibiotics actually can cause bacteria to become drug resistant. There are also rare bacteria that have drug resistance before exposure to the antibiotics, but either way, antibiotics are going to be able to select for mutations and things. And so the bottom line of this is, when you have an antibiotic, what you have to expect is resistance is absolutely going to be there. It's either already present and now you're going to start selecting for it, or mutations are going to start to come up and then you select for it. But antibiotics are a classic kind of selective pressure. So, you can think of this as evolution on a micro scale, is that those guys that are resistant to the antibiotic, in the presence of the antibiotic, they're off to the races and they're going to leave their sensitive brethren behind. Without the antibiotic being placed onto them, they may actually not even thrive all that well, but they certainly aren't going to be selected for. Now, genes can move around. So you can have an antibiotic resistance gene, this is a good one to talk about, that can actually jump about. But we can think about this movement in a couple of different ways. We can have within a cell something known as transposition, in which we've got kind of a copy and paste event occurring using something called transposase, an enzyme that binds to these little sequences that are on the transposon. It will loop out a piece of DNA, and then it can put it back in at a target site. It's very much like copy and paste. And you can either do a cut and paste or a copy and paste. And this is a way that genomes can actually evolve fairly quickly. We have the evidence in our genomes of transposition having occurred. Happily, we don't have any active transposons. Transposons, not surprisingly, can wreak havoc on a cell because they're happening in individual cells. They can disrupt the DNA. You can actually shatter the DNA, break it up. They can interrupt genes. Transposons are fairly nasty little guys. But we don't have to worry about it because we don't have any active transposons. Bacteria, on the other hand, some of them do. Other methods for gene transfer between bacteria, we've kind of talked about this. We've got what we call vertical transmission. We've done this already, in which we're talking about chromosome replication, followed by cell separation of the DNA, followed by cytokinesis, the pinching off the two cells. You make two cells. This is cloning. Now, horizontal gene transfer is some interesting things. You either have the uptake of naked DNA, which we were seeing in the Oswald-Avery experiment, where you had cells that were competent and being transformed by what he called the transforming factor, which is DNA. Transformation is a technique that we use in the lab, in the cloning lab all the time. We'll take DNA that's in the form of a plasmid and we'll pop it into cells. We'll transform them. Transformation requires very specific conditions, but it works fairly well once you have those conditions set up. Transduction is the term we use for the transfer of DNA by viruses. Now, the Hershey-Chase experiment was demonstrating this, in which they had the bacteriophage that were injecting their DNA. This is another way you can get DNA transferred from one bacterium to another, not through binary fission. And then finally, there is bacteria mating. It's called conjugation, which the bacteria have to be very close to each other. They create a conjugation tube, and DNA is transferred. It's basically mating between two cells. So, if we think about this, when we're talking about vertical transmission, we can create new alleles or mutations, that is, new forms of a gene, and they will be carried out in perpetuity to all the individuals. Now, this cell can rupture, DNA can be released, and we can have DNA being taken up, and that would be horizontal gene transfer. Now, horizontal gene transfer is going to result in a lot more mixing because you're not actually making a clone, you're adding DNA to an existing cell. And all kinds of things can happen with horizontal gene transfer. It's not nearly as clonal as vertical gene transfer. So horizontal gene transfer, as we mentioned, can kind of happen in a couple of different ways. We can have the release of DNA and have transformation, in which we've got a donor cell and a recipient cell, and the antibiotic resistance gene can be passed that way. We have a phage-infected donor cell that can be lysed. The phage can pick up the bit of DNA, and then they can, in turn, infect and transfer the DNA to a recipient cell. And then we have bacterial conjugation, in which we have a transposon can move a piece of DNA onto a plasmid that itself can be directed across. So these are some multiple ways of horizontal gene transfer: transformation, transduction, and conjugation. So transformation we were looking at by the Avery experiment, in which we've got the bacteria that has the ability to kill mice. Actually causes pneumonia. If we boil the deadly bacteria, that causes the cell the cells to actually break apart and bust apart. So the bacteria has lysed, and the bacteria's cell wall is opened up. DNA can move around, and the other bacterium, the non-lethal bacteria, can take up the DNA, and they can then become mutant. Now, we talked about this at first. What is making the pneumococcal bacteria virulent is the presence of this capsule. It's a smooth cell, and it is going to inhibit the way the mice can interact with this, and it also provides some toxins. Excuse me, it generates pneumonia. The non-lethal form of the bacterium actually doesn't have a capsule. It's not candy-coated. It's because it's missing a gene for capsule production. If that gene is taken up, then the the defective gene actually can be swapped out through recombination. And now this bacteria that was in fact non-infective and non-lethal begins producing its own capsule, and it becomes lethal. So this is how we can see an example of horizontal gene transfer through transformation. Now, with transduction, this is going to be a transfer of genes by viral infection. And it's a similar kind of thing, is that the virus can carry the gene from one cell to another. So the virus infects the bacteria. Some viruses package, when they package themselves up, they actually end up with host DNA fragments within them. The virus then injects the DNA into another cell, and now that cell, through recombination, can actually generate a new piece of DNA stable in the genome. It goes like that. So, you may have actually heard the Nobel Prize in Chemistry this year went to the two women that characterized something called the CRISPR-Cas9 gene editing system, which is actually found in bacteria, and it is a viral defense mechanism. We'll talk more about it later. So conjugation, this is about as close as bacteria get to having conventional sex, but it's not the same as sexual reproduction, because you only have DNA going from one organism to the other. You're not going to be actually making a hybrid kind of thing. So it requires this, what's known as an F prime or an F plasmid, and the recipient is going to be F minus. They produce a cytoplasmic bridge. They actually have a connection, a little tube, and DNA can move around, so the F plasmid can be transferred. Once that happens, the bacteria break apart. The F plasmid is transferred, and in this way, all the ladies become dudes. In a way of thinking about this, everybody that is involved in it now has the F plasmid, and they're able to carry out the next level of conjugation. But conjugation requires an interaction of actual cytoplasmic bridge. It's not naked DNA. So in order to transfer DNA through a conjugation, you have to have a mutation that is known as the HFR gene, which is high frequency recombination. And this involves the transmission of chromosomal DNA from one cell to another. So you have a pore forming between the cells, and this creates a bridge in which allows the DNA to actually be transmitted from one side to the other. The F factor, which is going to be carried on a plasmid, is responsible for actually generating this physical conjugation. And so if you don't have the F factor, you can't carry it out. And so you can have an F plus conjugating with an F minus recipient, and now they both have the F factor. And you can also transfer a variety of different bits of DNA. If the bridge is broken by disruption, you can have partial amounts of DNA. And so you can end up with a variety of different recombination events. So we got physical conjugation, F factor transfer, and then HFR transfer. Slightly different mechanisms of it, but not all that critical. So one of the ways to think about this is how, as a question, how would drug resistance gene might move from one bacterium to another by transformation, transduction, or conjugation? And a really important thing is to think about is that antibiotics don't cause resistance. They actually are what generates resistance. They are selecting for it. So the antibiotic is not generating, I guess I said that wrong. Antibiotic is not generating resistance, not carrying out mutations. It is selecting for those mutations that exist. So a drug resistance gene might be formed from another gene and then selected for by the antibiotics. That is, in the case of streptomycin, there is a ribosomal protein that has to change amino acid sequence. And when it does, it now is resistant to strep. Unless you add strep to this streptomycin, to this mix, you're never going to select for it. And when you do, it's off the races. So antibiotic resistance is a naturally occurring phenomenon that is going to happen when you use antibiotics. There is no other way around it. So one of the things that's interesting to think about is that some of these different control systems we've talked about, like heat and sterilizing, those kill bugs dead. But antibiotics, by their very nature, they can do a good job with an individual, but over time, they are going to lose their effectiveness. It's just going to happen. So that's why we have to be very careful about how we prescribe and what kind of antibiotics we do prescribe, because they're limited. You can think of it as a weapon that has so many times it can be unleashed, and then after that, it's no longer effective. It's quite the interesting battle of mutation and selection and change that is going on between the fight against bacteria and the bacteria's response to that fight. All right, we're going to start looking at some diseases and things next week, but that's all I know for now. I'm working on exams. On Monday, we'll go over the exam and see what's happening. We're getting set for another exam, but that probably won't be for another 2 weeks, okay? Hey, I'm Dr. G. I'm out of here. Peace. Have a good weekend.